From 1 to 2. Lesson objectives. Understand the different stages of mitosis and their significance in the process of cell division. Identify the key structures and events that occurred during each stage of mitosis. Evaluate the importance of mitosis in the context of the cell cycle and overall cellular function. Introduction. Have you ever wondered how living organisms grow, repair themselves, or reproduce? The answer lies in the process of cell division. Simply put, cell division is the process by which a cell divides into two or more daughter cells. All living things, from the tiniest bacteria to the tallest redwood tree, depend on this fundamental mechanism for growth and development. So, let's dive in and explore the fascinating world of cell division. Cell division. Cell division is the process by which a single cell divides into two or more daughter cells, each with the potential to grow, develop, and carry out specific functions. It is a fundamental process in biology that is essential for the growth, repair, and reproduction of living organisms. Cell division can occur through two main mechanisms: mitosis, which produces two identical daughter cells, and meiosis, which produces four genetically diverse daughter cells. Mitosis. Mitosis is the process by which a single cell divides into two identical daughter cells. This type of division is essential for growth and repair in multicellular organisms, as well as for asexual reproduction in some single-celled organisms. Meiosis. Meiosis is the process by which a single cell divides into four daughter cells, each with half the number of chromosomes of the parent cell. Meiosis is critical for sexual reproduction, as it allows for the production of gametes. Characteristics of mitosis. Mitosis produces two genetically identical daughter cells from a single parent cell. The major purpose of mitosis is for growth and to replace worn-out cells. Mitosis occurs only in somatic cells, which are all the cells in the body except for the reproductive cells. There is no reduction in the number of chromosomes during mitosis. Importance. Growth and development. Mitosis is essential for the growth and development of multicellular organisms. It allows cells to divide and multiply, leading to the formation of tissues, organs, and organ systems. Repair and maintenance. When tissues are damaged, such as in the case of a cut or injury, mitosis allows for the replacement of the damaged cells with new, healthy cells. Asexual reproduction. In some organisms, mitosis allows for the production of genetically identical daughter cells, ensuring that the offspring are identical to the parent cell. Vocabs to remember. Cell. A cell is the basic unit of life and the smallest functional and structural unit of living organisms. Split chromosome. A chromosome that has undergone a structural change, resulting in the separation of the chromosome. Chromosome. A chromosome is a structure found in the nucleus of cells that carries genetic information in the form of DNA. Vocabs to remember. Centrosome. A centrosome is a small organelle found in eukaryotic cells, which functions as a microtubule organizing center. Spindle fibers. The spindle fibers are microtubules that separates the chromosomes into two daughter cells. Centrioles. Centrioles are cylindrical organelles found in eukaryotic cells that are involved in the organization of cell division. Stages of mitosis. Mitosis, prophase. During prophase, the chromatin in the nucleus condenses into visible chromosomes, which become visible under a microscope. The centrosomes, which are responsible for organizing the spindle fibers, move to opposite poles of the cell. The nuclear envelope breaks down, allowing the spindle fibers to attach to the chromosomes. Mitosis, metaphase. During metaphase, the chromosomes line up along the equator of the cell, called the metaphase plate. The spindle fibers, which are attached to the centromeres of each chromosome, ensure that the chromosomes are properly aligned and can be separated correctly. Mitosis, anaphase. During this stage, the sister chromatids of each replicated chromosome separate and are pulled towards opposite poles of the cell by the spindle fibers. This results in the formation of two identical sets of chromosomes at opposite poles of the cell. Mitosis, telophase. During telophase, the chromosomes reach the opposite poles of the cell, and the nuclear envelope begins to reform around each set of chromosomes. The chromosomes begin to uncoil and return to their normal, diffuse form. The spindle fibers disassemble, and the cell begins to prepare for cytokinesis. Mitosis, cytokinesis. Cytokinesis is the process by which the cell divides into two daughter cells. In animal cells, a contractile ring of actin and myosin filaments forms around the cell, contracting and pinching the cell membrane until the cell is divided into two separate cells. In plant cells, a structure called the cell plate forms along the equator of the cell, eventually dividing the cell into two separate daughter cells. Mitosis is not just a process of cell division. It's a beautiful dance of chromosomes that ensures the continuity of life. Thank you for listening. Like, subscribe and press the bell icon to stay updated.